How's it going, everybody? So in this video, we're going to go over the eye anatomy and physiology. I'm going to start off by going through the parts of the eye. Then we're going to talk a little bit about how the retina works. And then we're going to go through some of the physiological activities you could do to test a lot of these structures that you find in the eye, just to see how they work. All right, so starting with the structures of the eye, I'm going to focus on the internal structures. All right, so this is a very rudimentary drawn eye. But we could see everything here, and the eye is really made up of three major layers. We have a fibrous outer layer, we have a vascular middle layer, and then we have the inner layer, which is the retina, which is made up of two different types of tissue. There's an outer layer that is full of melanocytes that's really used to absorb light, and then we have an inner layer of that is more neural tissue. Okay, so the major structures of those three layers. So the outer layer is mostly made up of sclera, which is a white fibrous tissue, and it's really there to hold the shape of the eye, and it's a good place for muscle attachment. And sclera in Greek means hard, and it is a more tough substance compared to a lot of the other structures in the eye. And this helps hold the shape of the eye a lot. So sclera means hard in Greek, and it is just a hard tissue that's used to keep the shape of the eye. The middle layer is mostly made up of your choroid, which is a blood vessel rich layer, and it's really bringing a lot of nourishment into a lot of these outer layers, or a lot of the tissue in the outer layer. You also can find melanin in here to help prevent light scatter. So it does help out a little bit with preventing too much light from bouncing around inside your eye. All right, then we have our retina, which is the inner layer, and that is made up of two layers in itself. So the outer layer of the retina is mostly melanocytes, and that's to help reduce light scatter, so you don't have too much light bouncing around inside the eyeball. And then we have a neural inner layer, which is all the different neurons that are being used to interpret the light that's coming into your eye. And this is where we're going to find the photoreceptors. So remember, we have two different types of photoreceptors. We have rods and we have cones. Rods are mostly going to be used for detecting dim light, while cones is more for colored light. All right, so going back to the outer layer of the eye, the majority of it is this sclera, this white connective tissue. Then we have this clear connective tissue that is down here. This is a continuation of the sclera called your cornea. And your cornea is clear to allow light to enter into your eye. The cornea itself does not have any blood vessels in it. And because of that, it still needs nourishment, but it doesn't get blood. So instead, it's going to use aqueous humor, which is this liquid that is right behind the cornea. So this is really where the nutrition is going to come from for the cornea and for the lens itself, which is deeper into the eye. So you have this really liquidy substance here that's bringing nourishment. But the aqueous humor also helps keep the pressure in your eye. After we have the aqueous humor, then we're going to find our iris. With this diagram, I have separated the iris and this ciliary body. This is not how it's going to look like in your eye. I did this just for the diagram so you clearly see these structures, but instead of all this stopping right here, imagine this continuing on to the edge or to the anterior part of the eye. So all of these push together up here. Again, I just separated these out just for the diagram. With the iris, this is going to house all the muscles that are used to open and close the pupil, which is that dark circle that's in the middle of your eye. So this is really what's opening and closing. So you have two different muscles in here. You have a sphincter pupillae and a dilator pupillae. And these are circular muscles that are sphincter is going to close and shrink the pupil. And then you have your dilator, which is going to open or expand and open the pupil. And then here's your pupil, and your pupil is really nothing but a window. It's not a structure. There's nothing there. The reason it looks black is because your eye is so good at catching scattering light that no light exits the eye. So there's no light escaping. That's why it looks black. It's not actually black. There's just nothing there. Now, the word pupil actually comes from Latin for pupillae or pupilla, which means little doll. And so the name came from when you stare into somebody's eye, you see your reflection at the pupil, and it kind of looks like a smaller version of yourself. So that's really where the name's coming from. So pupil is coming from pupilla, which means little doll, and it's talking about the little doll that you see when you look at your reflection in somebody's eye. All right, so the pupil is mostly just letting light in. It's controlling how much light comes into your eye because you don't want too much light in all the time. It's not doing anything to focus light. All right, the structure that changes to actually focus an image onto your retina is the lens. All right, so here is your lens right here. This will change shape. All right, it could be very bulbous and very round on each side, or it could be flat. And it's going to change depending on if you're looking at an object up close or far away. We call that accommodation. I'll talk about that later on in the video. And that's going to help focus light coming in so it hits your retina correctly. All right, what is controlling your lens or the shape of your lens is your ciliary body, which is this structure that surrounds the lens. All right, I have it cut in half here, but it should be a big circle around the lens. And this is an extension of the choroid. All right, so your ciliary body is part of this middle layer, the choroid. So the first part of the ciliary body is the ciliary muscle. I don't have it drawn here, but it will contract or relax. And between those two things will help open and close this lens. I'm going to talk about how all that works later on in this video as well. Your ciliary body also has ciliary processes, which are these processes that are sticking down here. All right, and these little projections here are going to hold the ciliary zonules, which are these fiber-like structures, these strings that are coming down from the process to the lens. All right, so as your ciliary body, as the muscles in it contract or relax, you're going to have these strings either tighten or get slack. All right, right behind that, we're going to have our ora serrata, which is really just a junction that's marking the end of the retina and separating the end of the retina to the ciliary body. So it's just a separation between retina and ciliary body. And then we have our vitreous humor. So this is another liquid-like substance that is in your eye. So with the anterior and posterior parts of the eye, the cutoff is the lens. So everything that's anterior of it, the liquid we find there is the aqueous humor. Well, the liquid on the posterior segment of the eye is your vitreous humor. This is more of a jelly-like substance. It is not liquidy. It looks like jelly. And this is really there to help hold the shape of your eyeball. All right, then we have special parts of the retina we can talk about. So near the back of the retina here, you're going to find a structure called your macula luteae. And this is where you're going to find a high concentration of cones. So there is some organization to the photoreceptors you have in your retina. Towards the back, near this macula luteae, as you get closer and closer to it, you get more and more or higher and higher concentration of cones. And the opposite is true as you go to the periphery of the eye. You get more or a higher concentration of rods as you go this way. So more cones this way, more rods this way. So the macula luteae is mostly cones. There's more cones than rods in this little structure right here. And in the middle of the macula luteae, you have another structure called your fovea centralis. And if you might remember from the bone chapter, fovea means pit. So you have this little pit that's inside your macula luteae called your fovea centralis. And inside the fovea centralis, you find nothing but cones. That you won't find any rods there. And then back here we have our optic nerve, which is taking all this information from your retina towards your brain. And if you notice, we have this retina coming down here, retina coming down here. It's all funneling towards the optic nerve. And at the optic nerve, we don't have any retina really. All right, we have this blind spot where we're not detecting light. All right, we call this our optic disc. So it's the circle that's in the back of the retina where the optic nerve is leaving, where we do not have any photoreceptors. Okay, so now we're going to look at retina physiology. So we're going to zoom into the retina, and we're going to see how it detects light and how it sends that information to the brain. But first, let me label everything here, and we can orient ourselves. On the left-hand side is more towards the anterior, so that's towards the pupil, while on the right-hand side is more towards the posterior, towards the back of the eye. And we have these three major cells that make up the neural layer of the retina. So we have ganglion cells, bipolar cells, and photoreceptors. The photoreceptors are what's going to be stimulated when light hits it. And we have two types of photoreceptors. We have rods and we have cones. So rods is more used to detecting dim light, while cones are more used for detecting colored light. Once they're stimulated, they're going to pass that information off to the bipolar cells, which are then going to pass it off to the ganglion cells, which is going to send that information down their axon to the optic nerve, where it's going to leave the eye and head towards the brain. And if you notice, the organization of this is kind of backwards to what you would expect. You would mostly expect the photoreceptors to be towards the anterior section of the eye, since that's where light is coming in. But that is not how vertebrates' eyes are set up. So cephalopod eyes, which is something like an octopus or a squid, they do have their photoreceptors in the front. And because of that, they don't have a blind spot like vertebrates do. You notice here how vertebrates' eyes are set up because the ganglion cells are in front sending information to the optic nerve and the photoreceptors are more towards the back. We have this area where there is no photoreceptors, where the optic nerve is leaving. So this is what we consider our blind spot. This is what the optic disc is in our retina. Alright, so let's try a little experiment to see if we could detect where the blind spots are in our eyes. So I'm going to draw this X right here, and then I'm going to draw a circle right here. Now, this might be different for everybody. There's a lot of variation between individuals as far as how big your blind spot is or where it's located in your eye. And everybody might be using a different size screen, so just keep that in mind. You might have to work a little bit to find your blind spot. But what I want you to do is to close your right eye and look at the circle with your left eye. Now, while you're looking at the circle, slowly bring your head closer to the screen. And at some point, you're going to see the X disappear in your peripheral vision. And it's going to disappear because we don't have photoreceptors in that part of the eye that's supposed to detect the X. For me, this is about 2 feet away from the screen, so it might be different depending on what screen you're using, and everybody's blind spot's a little different. But go ahead and work at it. And then you can do the same thing to your other eye. Go ahead and close your left eye, look at the X, and slowly bring your head closer, and at some point, the circle is going to disappear in your peripheral vision. Alright, so why don't we notice this during the day? Why don't we notice these blind spots? Well, because each eye is able to compensate for the other one. So that little area where you can't see in one eye, your other eye can detect, and you really don't notice it as you go throughout the day. Next, we're going to talk about real image, and what we mean by real image is the actual image that is projected onto your retina after it passes through the different structures of your eye. And this is caused by the refraction that goes on with some of these structures. So by refraction, I mean how light is deflected as it passes through different medium. And we usually measure this by a refractive index. So the structure light needs to pass through is your cornea, your aqueous humor, your lens, and then your vitreous humor before it finally hits your retina. And with the cornea, the aqueous humor, and the vitreous humor all have the same refractive index, so the light doesn't change too much from one to the other. However, the lens can change its refractive index depending on if it's bulgy or if it's flattened. So your lens is constantly changing its refractive index to help focus the image onto your retina. Now, as light passes through all these different media, the result is an image that hits your retina that is upside down, backwards, and smaller than the actual object. So all this refraction that goes on through these different structures causes this image to be reversed, inverted, and smaller. Now, we don't interpret it as upside down and backwards because our brain is able to correct this. Okay, so next we're going to talk about accommodation, and accommodation is how your lens changes shape in order to focus on near or far objects. And this has to do with the light coming off of that object. So typically anything that is 20 feet or more away, the light that actually hits your eye that had bounced off that object is mostly parallel with each other. Versus the light that bounces off an object that is closer to 20 feet, you still have some angle to the light rays that are bouncing off that object, and so your lens has to correct for that. So what happens is your lens, when it's focusing on something that is 20 feet or more away, needs to be completely flat because the light is already parallel coming into your eye. Versus objects that are closer, you need to add a kind of a bulge to your lens in order to redirect that light so it is focused on your retina. And I'll explain more of this in a second. But let's talk about the physiology of the changing lens, because it's not quite that intuitive if you're just seeing this for the first time. Alright, so this has a lot to do with the ciliary muscle that's in your ciliary body. Now remember, the ciliary body is a ring that goes around the entire lens. And it has the ciliary muscles and it has ciliary processes. And on the process, you have these ciliary zonule that go from the process to the lens. Now what happens is as the ciliary muscle contracts, it is going to shrink the ciliary body. All right, it's going to make it tighter. Versus when the ciliary muscles relax, it's going to open the ciliary body, or expand that ring, the ciliary body ring that is around the lens. So it's going to get wider as the muscles relax. All right, so contracting muscles, ciliary body tightens. Relaxed muscles, ciliary body opens. Now what happens is these zonules, these strings, are going to be either tight or loose, depending on if you have an open or closed ciliary body. So when the muscles are relaxed and the ciliary body is completely open, you're going to have tight ciliary zonules. Versus when the muscles contract and shrink that ciliary body, you're going to put some slack or make those zonules looser. Now with the lens itself, it's elastic and its normal shape, the shape it naturally wants to be in, is bulgy. Alright, so it wants to be wide. So when the ciliary muscles contract and shrink that ciliary body and put slack on the zonules, the lens is allowed to take its natural shape, which is a bulgy shape. Versus when the ciliary muscles relax and open that ciliary body, then the lens is going to be stretched flat because all these zonules are going to be tight now, stretching the lens out. As we get older, our lens actually loses elasticity, and that's part of what contributes to poor eyesight as you age. And we have a term for this. We call it presbyopia, which in Latin translates to old man vision. Okay, so let's see this in action by looking at an object that is 20 feet away. Alright, so remember, with objects that are 20 feet away, you want your lens to be flat. So for that to happen, the ciliary muscles will relax, expanding the ciliary body, which will tighten the zonules and then stretch the lens out. And the reason for this is because light that bounces off an object that's 20 feet away, you're going to have light scattering all different directions. And we're far enough away where the light that has an angle to it will scatter off into the environment somewhere, so the only light that is hitting your eye is parallel. Now let's compare this to an object that is closer than 20 feet. So let's say our lens is still flat and we're looking at an object that is closer than 20 feet. What's going to happen here is that the object is close enough where not all the light that is on an angle scattering off into the environment. That angled light is actually entering your eye. And if your lens is still flat, that light's going to be at an angle when it hits your retina as well. So it's going to look blurry. So to accommodate this, your lens is going to change its shape. And again, it's going to do this by first tightening the ciliary muscles, which is going to shrink the ciliary body, putting slack to the zonules, allowing the lens to take its natural shape, which is bulgy. And this bulginess of the lens can then take that light that's coming off at an angle and redirect it so it is focused when it hits your retina. Okay, so let's talk about some visual problems that could occur. Now, there's a lot of factors that contribute to your eyes functioning properly, and one of them is the shape of your eye itself. Now, when an eye is properly shaped, the light that enters the eye and refracts through the lens will have a focal point that lands on your retina. So when we have a properly shaped eye, we have a term for this. We call it emmetropic. But what if the eye isn't properly shaped? What if the eye is too long? Well, we have a term for that. We call it myopic, or being nearsighted. So with the myopic eye, what happens is as light enters the eye and refracts through the lens, the focal point is going to land before the retina. So what the retina detects is after the focal point where the image is blurry. And what about an eye that's too short? Well, we call that hyperopic or farsighted. And with a farsighted eye, as the light enters the eye and then reflects through the lens, the focal point is going to land behind the retina. So what the retina is picking up is before the focal point. And again, it's going to be blurry here as well. All right, let's take a look at how we could correct these vision problems. Again, with myopic eyes, they're nearsighted, so that means the focal point is landing before the retina, and hyperopic is farsighted, where the focal point is landing after the retina. And we want them both to function like the emmetropic eye. So with the myopic eye, we have this focal point before the retina. Well, one way to bring that focal point back towards the retina is to increase the angle of the light coming into the eye. So if we increase the angle, that means the focal point is going to fall further behind. So one way to do this is to use a concave lens. So whether it's glasses or contacts, if we add concavity to the lens, the light is going to follow that concavity and expand its angle coming through the lens. So now we have a wider angle that hits our eye. So since we have a wider angle hitting the eye, that means the focal point is going to land further behind. So with the hyperopic eye, we have a focal point that's landing behind the retina. So to correct this, we want to make sure the angle of the light coming in is smaller. And to do this, we use a convex lens. And again, the light is going to follow the convex part of the lens, meaning the angle of the light coming in the eye is going to be smaller. And that's going to bring the focal point forward to where it hits the retina. Okay, so how could we tell if somebody has less than average vision? Well, one way to do it is to use this chart, and if you've ever been to the doctor, I'm sure you've seen this chart before, but it has a name. It's called a Snellen eye chart. And how this chart works is that the subject stands 20 feet away from it and then reads line by line with each eye until they can't read anymore. So let's say you could read up to this mark right here. This is the 20/20 mark. And what that means is that at 20 feet away, you're reading what the average person can read at 20 feet away. But not everybody has average vision. Some people have worse. So let's say that you could only read up to this line. This is the 20/30 mark. And what that is saying is at 20 feet away, you're reading what somebody with normal eyes could read at 30 feet away. So somebody with average vision could back up 10 more feet and read this line as well. Some people have better than 20/20 vision. Some people could read up to this line. This is your 2015 mark. And that is saying at 20 feet away, you're reading what somebody with average eyes would have to stand at 15 feet away to be able to read. So the Snellen eye chart is a very common way of testing how well somebody is able to see. So having incorrectly shaped eyes is not the only way to have poor vision. Something else that could happen is having irregular curvature inside your cornea or your lens. And this could also cause blurriness. So when this happens, we have a name for it. It's called astigmatism. And you may have seen this chart at the doctor's office as well. This is called an astigmatism chart. And how this works is that you look directly in the center of the chart with each eye. And if any of the lines look blurry or lighter in color, there is a good chance that you have an astigmatism. However, if all the lines are uniform as you're staring at the center, usually means that you don't. So astigmatisms can also be corrected with glasses. So when they're making the lenses to the glasses, they could add what is known as cylinder and axis to the glasses. What cylinder is going to do is correct for how strong the astigmatism is, while axis is going to correct for the position of the astigmatism. Okay, so now we're going to talk about the visual pathways that lead to the brain and how your brain is interpreting the images that your eyes are detecting. So this picture is of the inferior view of a brain. So we're looking at the bottom of somebody's head. So we're looking underneath. And you might remember this structure that's right in the middle called the optic chiasma. So this is a structure that we covered in the brain chapter. And this is where some of the tracks from your optic nerves are going to cross each other. Alright, so since we're looking at the inferior view, remember on your left-hand side is actually the subject's right, and then on your right-hand side is the subject's left. And the interpretation of vision is going to happen in the occipital lobes of the brain, so all the way in the back of the brain. And specifically, the right occipital lobe is what is going to be responsible for interpreting left field of view, while the left occipital lobe is going to interpret the right field of view. Alright, so I'm going to draw the right field of view with blue and the left field of view with red. And so we're going to start with the right eye first to understand what it detects. So the right eye is going to see most of the right side of your field of view and a little bit of the left side. So here's everything your right eye is detecting. So we have the right part of vision in the blue and the left part of vision in the red. And then your left eye is going to detect more of the left side than the right side, but it also detects a little bit of the right side. And then you have this area overlap between the two of them, and this is what gives you your binocular vision. I'm going to draw the binocular vision in purple. Alright, so let's see how the light enters the eye, because it'll help make sense of how it's traveling down these tracks out of the optic nerve to your occipital lobes. So let me write this real quick. So right eye is seeing that, left eye is seeing that. Alright, so let's go ahead and start with the right eye and see how the light is entering the eye. Now, pulling everything we talked about together, remember the image that you see is reversed as it comes in. So everything on the left part of your right eye vision, so I'm going to go ahead and draw this line here. You see all this light's going to come into your eye, and it's going to reverse, so the image is going to hit more of the right section of your retina. Well, in the middle is going to be closer to the middle of the retina. And then the right side is going to come through the pupil and hit the left side of your retina. Remember, the image is reversed. So let me draw the parts of the retina that are actually seeing this. So here is where all the right vision is being brought in. Here is where all the left vision is being brought in, or hitting the retina. But again, our left occipital lobe needs to interpret everything in the right field of view, and the left field of view needs to be interpreted by the right occipital lobe. So how does this work? So as the axons leave the optic nerve, everything that was in the right field of view, the axons that detected that stimulus, they're going to cross through that optic chiasma and head towards the left occipital lobe, because the left occipital lobe needs to interpret the right field of view, or the right vision. While everything that is on the right part of the retina that detected the left part of the field of view, those axons are going to travel straight back towards the right occipital lobe. And we could do the same thing for the left eye and what the left eye is detecting. So we have this light coming in. It's going to hit more towards the right side of the retina. Then we have the center hitting more towards the center of the retina. And then we have the right side of the field of view for your left eye hitting the left part of the retina. So let me go ahead and draw the parts of the retina that are seeing right and left. So everything in blue is seeing the right field of view. Everything in red is seeing the left field of view. Now everything on that left field of view that detected the left field of view needs to be interpreted by the right occipital lobe, so those axons are going to travel across the optic chiasma, head towards the right occipital lobe. And everything that detected the right field of view is going to travel straight back towards the left occipital lobe. So now everything that we had that was in the right field of view is now being detected by left occipital lobe, and everything that we saw in the left field of view in both eyes is going to be interpreted by the right occipital lobe. Alright, so that's everything I wanted to talk about in this video. I'm going to upload other videos of a sheep eye dissection and going through a human model of an eye to help with identifying some of these structures.